Assalamu alaikum class. Today we will be studying about the sex determination and sex-linked inheritance part 2. In this video, we are going to talk about sex-linked and sex-influenced inheritance. Before starting our second part of the sex determination and sex linked inheritance, let me just briefly explain to you what we have studied so far in the previous part of this series of lectures. We have learned different systems of sex determination, and through these systems, we have learned that how in different organisms, such as in insects, grasshoppers, flies, amphibian mammals, including the humans, butterflies, and even in some plants, fungi, and protozoans, how the sex is determined, which system is present, and which mechanism is responsible for the formation of the males and the females. And we have also learned about the mechanisms in which there is no distinct sex chromosome, and the sex is actually determined by the undifferentiated chromosomes. So in today's discussion, we start with Drosophila melanogaster, how sex is determined in this, and what are the sex-linked inheritance pattern in case of Drosophila melanogaster. As we have already studied that the fruit fly, Drosophila melanogaster, it has 8 chromosomes, 3 pairs of autosomes and 1 pair of sex chromosomes. Thus, it has inherited one haploid set of the autosomes and one sex chromosome from each of its parents. Normally, female flies that have two X chromosomes and males that have an X chromosome and a Y chromosome. But the presence of the Y chromosome does not determine maleness in case of Drosophila. Instead, each fly's sex is determined by a balance between the genes on the autosomes and the genes on the X chromosome. This type of sex determination is called the genic balance system. In this system, a number of different genes influence sexual development. The X chromosome contains genes with the female producing effect, while the autosomes contains genes with male producing effect. Consequently, a fly's sex is actually determined by the ratio of the sex chromosome, which is the X chromosome, to the ratio of the autosomes. The number of the X chromosomes, which is then divided by the number of the haploid set of the autosomal chromosome. So if the ratio is X ratio A, which means that the ratio of the X chromosome to the autosome is 1, then it's going to produce a female fly. And if the ratio of the X chromosome to the autosome is 0.5, it's going to produce a male. In this table, it is actually summarizing the ratios and what would be the sexual phenotype of the living organism. Now, to just understand how these ratios are deciding the sex of the phenotype of the fly, let's review this in a video in the next slide.
The fourth type of determination system that we're going to talk about is very specific to Drosophila, or the common housefly. So, just to give you a little bit of background, Drosophila has a total of 8 chromosomes, and those chromosomes really split out into 4 pairs. So, 3 pairs of chromosomes are going to be autosomes, and 1 pair is going to be sex chromosomes. The female is going to be have the genotype of XX, and the male will be XY, but it's very different than the human system. Y actually does not determine the degree of maleness in Drosophila species. So you'll see what we're talking about in just a few minutes. So the way that they actually came up with this hypothesis was they worked through the scientific method to make these conclusions. Their original hypothesis was that the fly's sex was actually determined by a balance between the genes on the autosomes and the genes on the X chromosome. So the X chromosome would contain genes that had female effects. So they would code for protein that were very specific to the female phenotype. And then they hypothesized again that the autosomes would actually contain genes that would code for proteins specific to male effects. And so what the result of those two hypotheses was that the fly sex was actually predicted by the X chromosome to autosome ratio. So X to A is the X chromosome to autosome ratio. So, more current research had actually determined that genes on the X chromosome are the sex determinant. Okay, so they influence the autosomal chromosomes a little bit, and so the effect of those autosomal chromosomes are very indirect when compared to the genes that are being expressed on the X chromosome. So what ends up happening is there's a control of expressional timing. Okay, so there's going to be a very specific time when the X chromosome genes are actually going to be expressed and upregulated to go through transcription and translation. So, as we take a further look at this genic balance system in Drosophila, um, there is the prediction that the number of X chromosomes is going to be in, if we take the number of X chromosomes and divide by the number of haploid sets of autosomal chromosomes, then we get the sex of the Drosophila or of the housefly. So if we have an X to A ratio of 1% or 1.0, then that individual will be female. However, if we have an X to A ratio of 0.5, then that individual will be male. So as you can see in this figure here, we've got our female fly. She is going to produce um, these sets of autosomes, and so set number 1 of autosomes, set number 2, and set number 3. And then she's also going to produce one set of sex chromosomes. Okay, so in our male species, they're also going to produce 3 sets of autosomes, but the sex chromosome is going to have a very different Y chromosome compared to the X. All right, so I encourage you, if you're having trouble following the Drosophila logic, to look at Table 4.2 and actually read this section in more in depth in your book. And this will actually help you be able to determine what the sexual phenotype of an individual fly is based on the ratio of their sex chromosomes to their autosomes.
Sex determination in humans. Humans like Drosophila have XX and XY sex determination. But in humans, the presence of the gene, which is called as SRY gene, on the Y chromosome determines the maleness. The phenotype that results from the abnormal number of the sex chromosome, which arise when the sex chromosome do not segregate properly in meiosis or mitosis, illustrate the importance of the Y chromosome in human sex determination. Now, the first example that we are going to discuss, which is because of the improper segregation of the sex chromosome during the formation of the eggs or the sperms, is Turner syndrome. So on the left side you see that a female who is actually affecting from Turner syndrome, and on the right side you see the karyotype of that female. So the Turner syndrome, the person who are actually suffering from this syndrome are females, and they often have underdeveloped secondary sexual characteristics. This syndrome is seen in 1 out of 3,000 female births, and the affected females are frequently short, and they have low hairline and relatively broad chest and folds of skin on the neck. Their intelligence is usually normal, and most women who have this Turner syndrome are sterile. In 1959, Charles Ford used a new technique to study human chromosome, and he discovered that in the cells of a 14-year-old girl who was actually suffering from the Turner syndrome, she had only a single copy of the X chromosome. As you can see on the right side, you see the karyotype of this female. She has the normal 22 pair of homologous autosomes, and you see one X chromosome, and the other X chromosome is actually missing here. So you can say that the chromosome complement is actually referred to as XO, having just 45 autosomes and one X chromosome, one sex chromosome, and the other X chromosome in case of females is actually absent. Another example of improper segregation of the sex chromosome during the process of meiosis or the formation of the gametes is Klinefelter syndrome. Persons who have Klinefelter syndrome, which occurs like with a frequency of 1 in 1000 male births, have cells with one or more Y chromosome and multiple X chromosome. The cells of most males having this condition are XXY, but the cells of few Klinefelter males are XXXY, or even they have 4 X chromosome and 1 Y chromosome, or they may also have 2 X chromosomes or 2 Y chromosomes. Men with this condition frequently have small testes and they reduce to produce facial or pubic hair. They are often taller than normal and sterile. Most of them have normal intelligence. Another example of improper segregation of the sex chromosome, which occurs normally in the females, is called as poly-X female. Now, these poly-X females, these females, since they have three X chromosome, as you can see in the karyotype on the right side, they have three X chromosome instead of having a normal set of X chromosome having just two X chromosome. These are also sometimes referred to as super females. Now, poly-X females, which occur in about 1 in 1000 female births, the infant cell possesses 3 X chromosome, and the condition is also sometimes referred to as triplo X chromosome or the triplo X syndrome. Now, these persons have no distinctive features other than having a tendency to be tall and thin. Although a few are sterile, many menstruate regularly and they are actually fertile. Now, the incidence of mental retardation among the triple X female is slightly greater than in the general population, but most of the triple X females have normal intelligence. Much rarer are the female whose cell contain 4 or 5 X chromosomes. These females usually have normal female anatomy but are mentally retarded and they have a number of physical problems as well. The severity of the mental retardation increases as the number of the X chromosome increases beyond 3. The phenotypes associated with the sex chromosome anomalies allow us to make several inferences about the role of sex chromosome in human sex determination. For example, number 1, the X chromosome contains genetic information which is essential for both sexes. At least one copy of an X chromosome is required for normal human development. Similarly, the male determining gene is located on the Y chromosome. A single copy of this chromosome, even in the presence of several X chromosomes, produces a male phenotype. The absence of the Y chromosome results in a female phenotype. Genes affecting fertility are located on the X and Y chromosome. A female usually needs to at least two copies of the X chromosome to be fertile. Additional copies of the X chromosome may upset the normal development in both males as well as in females, producing physical and mental problems that increases as the number of the extra X chromosome increases. Now let's look at the male determining gene in case of human, which is also called as the sex determining region Y or the SRY gene. This male determining gene in humans, which is as I told you, is called the sex determining region on the Y chromosome, and it was actually discovered in 1990. This gene is found in the XX males and it is actually missing from the XY females, and it is also found on the Y chromosome of other mammals. Definitive proof that the SRY gene is the male determining gene came when scientists placed a copy of this gene onto XX mice by means of genetic engineering. The double X mice or the XX mice that received this gene, although they were sterile, they developed an anatomical male feature. The SRY gene encodes a protein called the transcription factor that actually binds to the DNA and stimulates the transcription of other genes that promote the differentiation of the testes. Although SRY is the primary determinant of the maleness in case of humans, other genes which may be also X-linked and some may also be present on the Y chromosome and some other may be present on the autosomes as well, also have roles in fertility and the development of sexual differences. Although the SRY gene is the primary determinant of the sex in case of human embryos as well, the other genes are actually influencing the sexual development. As you can see in the case of females with androgen insensitivity syndrome. On the right side, you see a Belgian supermodel who has this androgen insensitive syndrome, and you see that she looks perfectly normal. The cells of a woman with androgen insensitivity syndrome contain an X chromosome and a Y chromosome. They look like females, but they are actually not. Now let's look at some of the sex-linked characteristics, and you all know that alleles on the sex chromosomes are actually inherited in a predictable pattern. So the genes which are present on the Y chromosome, or which are the Y-linked traits, they can be inherited from the paternal grandfather, that is your father's father, and never from the maternal grandfather, since X-linked characteristics also exhibit a distinct pattern of inheritance. So we have three type of characteristics. They can either be X-linked, they can either be Z-linked, or they can be Y-linked. And in case of Y-linked characters, they exhibit a distinct pattern of inheritance, and they are present only in the males, and all the male offsprings of a male with a Y-linked trait inherit this trait. And relatively, I have to tell you that relatively there is little genetic information which is actually present on the Y chromosome. So first of all, let's look at a X-linked character, which is color blindness, very common in the humans. Let's just look at that. But before we start explaining the X-linked color blindness, I want all of you to look at this color test plate on the left side of this slide and check if you can see a number written inside it. So did you see something? If you see a number 74, congratulations, you have normal vision. You're perfectly normal. If you see number 21, I'm sorry, you are colorblind. But hey, if you don't see any number, I'm really sorry because you are totally colorblind. Still, you don't have to worry. It's not a big issue to worry about. Let me explain you how. Well, color blindness, as you know, it's also known as color vision deficiency. It is actually the diseased or less ability to see colors or differences in the colors. Simple tasks such as selecting ripe fruit, choosing clothing, and reading traffic lights can be more challenging. Color blindness may also make some educational activities more difficult. However, problems are generally minor and most people find that they can actually adapt to this situation. People with total color blindness, which are also sometimes known as achromatopsia, may also have decreased visual activity and they may be like very uncomfortable in the bright light. The most common cause of the color blindness is actually an inherited problem in the development of one or more of the three sets of the color sensing cones in the eye. Let me just tell you very briefly that our eye has two types of cells. We have rods and cones. Rods are the ones that let us see when it's a little dark, so they can let us see in the dark environment, whereas the cones are the cells that actually sense different type of colors. Now, the males are more likely to be colorblind than the females, as the genes which are responsible for most common forms of the color blindness are actually present on the X chromosome. As the female has two X chromosomes, a defect in one is typically compensated by the other. Therefore, females can be only carriers. Males only have one X chromosome and therefore express the genetic disorder. Color blindness is typically, as I told you, it's a inherited disorder, genetic disorder, and as I told you that it is commonly inherited from the mutation on the X chromosome. But the mapping of the human genome has shown that there are many causative mutations, and these mutations are capable of causing color blindness. It originates from at least 19 different chromosomes and 56 different genes. About 8% of the males and about 0.4% of females are red-green colorblind in some way or another, whether it is one color, color combination, or another mutations. Males are at a greater risk of inheriting an X-linked mutation, as I told you, because males have only one X chromosome and the other one is the Y chromosome, and the Y chromosome doesn't carry the gene to compensate the effect. And the females have two X chromosome, and the compensation occurs there because if she inherits like one normal X chromosome, this mutation is not going to like display itself. And as I told you, that men do not have the second X chromosome to override the chromosome that carries the mutation. So if 8% of the variants of a given gene are defected, the probability of a single copy being defective is 8%. But the probability that two copies of both of them are defected is 0.64%. Y-linked characteristics. As I told you, they are, since the Y chromosomes are present only in the males, so the Y-linked characteristics are present only in the males, and all the male offsprings will exhibit this trait. And the Y chromosome, actually, it carries very less number of genes, that it has lost most of the DNA over the time. And the most important thing which is present on the Y chromosome is the SRY gene. Now, before I explain the Y-linked characteristics, let's watch something really interesting, which is related to Y-linked characteristics.
From the nation that brought you the man who cuts hair with his mouth and the man who pulls vehicles with his moustache, India introduces the man with really long ear hair. The bloke attached to those extraordinary locks is Radhakant Bajpai. He's from the northern city of Kanpur, and if you're smirking at his unique look, then stop it, because growing awesome ear tufts is a serious business.
The government's not providing any assistance and society is apathetic. People just consider sporting long ear hair to be an act of foolishness. Had the government commended this feat, society would also have been quick to accord recognition.
Well, Indian society may not be ready for him, but the Guinness Book of Records certainly is. Read 'em and weep, ear hair enthusiasts. That's 13.2 centimetres and it's official. But there's more. That certificate is 5 years old. Radhakant's been smashing his own stat every day since. Today, his world-beating oral bush now measures almost 25 centimetres. And if you think his son Satyam's going to be embarrassed by his dad's style, think again.
I have never felt that ear tufts are odd because I'm used to seeing them. Once, when I was registering for college with my father, people stared at him, and then I realised it was something unusual.
But who wants to be usual when you can be a record breaker? And dedication's what he's got. Radhakant combs his tufts daily, he uses a special shampoo, and he's even stopped wearing jewellery to minimise potential damage. And when he's not looking after those record holders, he's a grocer in the local market. And fellow store owner Lakshman Gupta reckons Radhakant's hairy ears are great for business.
Our market's gained fame and recognition along with him.
Respect from your peers, respect from your family. If you want to be a success, why not do it the Radhakant way and be proud of that ear hair?
Some other sex-linked traits include Y-linked traits. So this is actually called hairy ear syndrome. So this is a gene that is on the Y chromosome, and we can actually see this gene being transmitted from fathers to sons because it can only go from Y chromosome to Y chromosome. The X chromosome doesn't have anything to do with the expression of this gene. So this gene really results in additional proteins being produced and additional hair follicles being produced in the ear area, in the ear areas. Sorry.
Now let's look at the Z-linked characteristics, and as we have discussed previously that in organisms which have this ZZ and ZW sex determination system, the males are the homogametic, which means they are producing only one type of gametes, both of them containing the Z chromosome, and the females are heterogametic, which means that they produce two type of eggs, one containing the Z and the other one containing the W chromosome. Now, same as that of the X-linked characteristics, except that the pattern of inheritance in the males and females is reversed, Z-linked characteristics in the cameo phenotype in Indian blue colored peacock is a very good example to explain the Z-linked characteristics. Let's watch how these characteristics are inherited from one generation to the other generation.
ZW sex determination system is a chromosomal determination system that we see in a lot of avian species. Okay, so just keep in mind that in the avian species, the female and the male are actually switched from what we know in the human species. So the females are going to be heterozygous here, and the males are going to be homozygous. So in our parent generation in peacocks, if we have a blue female, her genotype is going to be ZCA plus, and then she's also going to pack a W genotype as well. And we bred that to a cameo male, which is homozygous ZCA for the cameo phenotype. And then in the reciprocal cross, we get two very different looking individuals. So our Camino female is going to have shorter tail feathers than our Camino male. And then our blue male is going to have the really beautiful, long, colorful tail feathers, which our blue females do not have. So these Z-linked traits are really going to be expressed fully when associated with the male chromosome. So as we look at the F1 generation from the parents that we had a ZCA W individual and our dad was homozygous CCA, that resulted in 50% male and 50% female offspring. The male offspring ended up having the CA plus and just the CA alleles associated with the Z chromosome that resulted in the beautiful blue long-tailed males. And then the cameo female is going to be a carrier of the ZCA allele and the W. And then in our reciprocal cross, we get a blue male, similarly to what we saw in the previous example, and we get a blue female as well. But when we get to the F2 generation, that's where we get a lot of genetic variety in our phenotypes. So we can have a quarter of the individuals are actually going to be blue male, a quarter are going to be blue female, a quarter are going to be cameo male, and a quarter are going to be cameo male. And then the F2 generation, we actually have a 2 to 1 to 1 ratio. So we've got 50% of the offspring that are going to be blue males, and then 25% that are going to be blue females, and 25% that are going to be cameo females. So just keep in mind that the big letter, the Z here, refers to the sex chromosome, and then the small level letter that is a superscript, that really refers to the version of the allele for the color gene that these individuals have.
So now we have excellent characteristics, and there is a very important part that you need to look at, which is called as dosage compensation. You know that the females, human females, and most of the mammalian females, they have two X chromosomes, and during their lifetime, one of their X chromosome is actually you can say that it is inactivated or converted into a bar body. This process is called as dosage compensation, and this X inactivation, which is also called as lionization, which was actually because it was discovered by the English geneticist called Mary Lyon. It process by which one copy of the X chromosome is inactivated in all the mammalian females. The inactive X chromosome is silenced by like being packaged into a transcriptionally inactive structure called the heterochromatin. As nearly all the female mammals have two X chromosome, X inactivation prevents them from having twice as many X chromosome gene products as compared to male. And you know that the males have only one copy of the X chromosome. So that's why it's called dosage compensation. Now, the choice of which X chromosome will be inactivated is actually random in the placental mammals, such as in case of humans. But once an X chromosome is inactivated, it's going to stay inactive throughout the lifetime of the cells and its descendants in the organism. Unlike the random X chromosome inactivation in placental mammals, inactivation in the case of marsupials applies exclusively to the paternally derived X chromosome. Now, how and why this inactivation of the X chromosome takes place, let's watch that.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about X inactivation. Sometimes we call this lionization, and that's kind of giving credit to the person who discovered most of this, which is Mary Lyon. But I want to start the whole thing by talking about calico and tortoiseshell cats. Calico cats like this one and tortoiseshell cats like this are almost always female, which puzzled scientists for a long time until we finally figured out how that works. And it puzzled Mary Lyon as well. Here she is. She worked at Cambridge. But basically what she found is that when she was working with lab rats, some of them would be a white, some would be a gray, and some would be kind of a tortoiseshell between the two. But what she found is that most of those tortoiseshell mice were females, and so most of them had two X chromosomes. And she was also working on chromosomes and why some disorders are more prevalent in females than they are in males, and so we know that's sex linkage. But basically what she found is that when you look into the nucleus of a cell, so this is in the nucleus, in every cell of every female, one of the X chromosomes will be active, and so it works perfectly, but the other one is going to be inactive. It's going to fall apart. And so if you're a female, in all of your cells, only one of the X chromosomes is active and the other one is inactive. It's okay though, because you're fine. On guy's side, same thing. We only have one X chromosome, and so sex chromosomes like this are kind of designed to just work on their own. When it breaks apart like that, we call that a bar body, or that X chromosome is now inactivated. And so let me kind of talk you through how this works. Well, basically when you're born, before you're born, when you're a zygote, that zygote, if you're a female, will have two X chromosomes. And those X chromosomes will be copied when the cell goes through mitosis. And so we go from having one cell to two, and then to four, and then to eight. And I could keep doing this, but basically when you get a big ball of cells like this, these are called stem cells. They're cells that can become any other cell. But right around this time, when you start to get this big blastula, there's basically a flip of the coin that goes on in each of the cells. And so one of the X chromosomes will simply wad up and become inactivated. So this one might wad up, and it's a 50-50 chance which are the ones are going to wad up. So it might be that one, it might be that one. So it's totally random which one's going to wad up in each of these. Now, the one that's left, it's going to be able to express all of the genes that are found on that. And what's interesting is that all the cells that come from this cell likewise are going to have one X chromosome that's inactivated and then one X chromosome that's activated. And so basically this stem cell, once that flip of the coin has occurred, it's for the rest of the life of the cell and all the cells that come from that. And so that tells us why a calico cat is female. And let me talk you through that. So if you're a male cat, you could basically be an orange cat or a black cat. Or you could be a white cat if you're lacking the pigment on the hair. And so let's look at this. So it's found on the X chromosome. If you have the black gene on your X chromosome, since you only have one X chromosome, then you're going to be a black cat. If you have the orange gene on your X chromosome, then you're going to be an orange cat. And it's pretty straightforward because in every cell there's going to be an X chromosome with that gene. It's making that pigment color. Now let's look at the female side. So if you're a female cat and you have two X chromosomes, if both of them are black gene, then you're going to have a black cat. Now what you should know is that in each of the cells of this cat that produces this black pigment on its skin, it's going to be a different X chromosome that's working in each of those. So that flip of the coin has occurred. Likewise, if we've got an orange gene in each of those X chromosomes, same thing. It's going to be orange. But what's weird is if you're heterozygous for it. And so if you're a female who has one X chromosome with a black gene and one with a orange gene, well, how could this occur? Well, it's simple. Let's say we match a black cat with an orange cat, then all of the offspring are going to be like this. And so what is it going to look like? Well, this is not a great animation or a great picture, but basically what you'll have is splotches of black and splotches of orange. And so let's look at this black right here. Basically what happened is at the stem cell stage, the orange gene, this orange gene became a bar body, so it became inactivated, and all the cells that came from that produced black pigment. I think that's really cool. Likewise, in ones where the black one wadded up, all the cells that come from that is going to produce orange pigment. And so that's X inactivation. It's rare that we see this occurring, and that's just because sex cells are designed to work on their own, the X chromosome. Now, two quick questions you should be thinking right away is, number one, what about the white? Why do we get this white coloration? Well, if you remember when I talked about genetics, there's a process called epistasis. And so basically there's going to be a set of genes that determine the color of the pigment that's added to the hair, but there's going to be another gene or set of genes that are actually going to tell us, is that color added to the hair or not? And that's called epistasis. And so basically in these cells, we're not actually adding the pigment, but up here we've got X inactivation. Another question you should be thinking to yourself is, at the beginning I said almost all calico cats that you'll ever see are going to be female. Well, how could we ever have a male calico cat? It's pretty simple, but maybe not intuitive. Basically what you could get is a XXY male. And so how does that occur? Well, during meiosis, you could get non-disjunction of the two X chromosomes. And so basically you could get one that is of the orange, one that is of the black. And so you could have we call this Klinefelter syndrome in humans, but you could have a calico male. But again, it's going to be incredibly rare. So that's X inactivation. It's rare that you could see this in humans. However, I was reading, and there's a disorder in humans where they lack sweat glands. You could imagine that's really, really dangerous because you can't regulate your temperature. And what I found is that if you have or are heterozygous for this in some females, they'll actually have patches of their skin where they have sweat glands and patches where they don't. And that's just due to X inactivation. And I hope that's helpful.
All right, so now the summary of what that we have learned in this lecture of sex determination and sex-linked inheritance part 2. First of all, we learned about how the sex is determined in case of Drosophila, and we found that in case of Drosophila, each fly's sex is actually determined by a balance between the genes on the autosomes and the genes on the X chromosome. And I also explained that this type of sex determination is called as genic balance system. And in this system, a number of different genes which are actually influencing the sexual development. The X chromosome contain genes with the female producing effect, and the autosomes contain gene with the male producing effect, and therefore our fly's sex is actually determined by the ratio of the X chromosome to the autosomes, and the number of the X chromosome divided by the number of the haploid sets of the autosomal chromosomes. And then we also learned about the sex determination in case of humans, and we all know that like Drosophila, humans have this XX and XY sex determination. But in case of humans, the presence of the sex determining region or the gene SRY gene, which is actually located on the Y chromosome, is very important. It determines the maleness and the phenotype that result from the abnormal number of the sex chromosome, which arise when the sex chromosome actually do not segregate properly during the process of mitosis or meiosis. And we also discussed some examples. For example, we learned about the Turner syndrome, how this happens, and we discussed that it occurs. It's very rare. It occurs at like 1 out of 3,000 female births, and in the case of Turner syndrome, the females who are like, they look like females but they are actually not because they are sterile, they have one copy of this X chromosome instead of having two. And then we also learned about the Klinefelter syndrome, in which this normally happens in case of the males, in which they have an extra copy of the X chromosome along with an X and Y chromosome. So they can have two X chromosome and one Y chromosome, or three or four, or even and they can have like five number of X chromosome and one Y chromosomes, and sometimes they can have like multiple number of like more than two or more than two Y chromosome along with multiple number of the X chromosome. And with the increasing number of the X and Y chromosome, their mental ability is actually being affected. And then we learned about the poly X females. Poly means many, and X means they are having like a lot of number of the X chromosomes. And the ratio of them, or the frequency of the woman being born with many X chromosomes, is 1 out of 1,000 births. Then we also studied about the some of the sex-linked characteristics, like we learned about the X-linked, Z-linked, and the Y-linked characteristics. In case of the X-linked characteristic, we studied about an example of color blindness, and it can like normally affect the males because they do not have another X chromosome to compensate the effect of this mutated gene which is present on one defected X chromosome. Then among the Z-link characteristic, we learned about the example of the feather color in case of the peacocks. And in case of the Y-link characteristics, we learned about the hairy ear syndrome, in which the male is actually transmitting this gene which is present on the Y chromosome to all his male offsprings. In case of then finally, in case of the X chromosome inactivation, we learned about the Barr bodies, or the phenomena of lionization, and we also studied the example of the tortoise shell cats, and we understood that how and why it occurs only in the females. Okay, so since we have covered our chapter on sex determination and sex linked inheritance pattern in part 1 and today in part 2, there are some questions that we are going to discuss in our class after the completion of this lecture. So what I'm going to ask you, and what you have to study about, you have to learn about that, and then we are going to discuss that, and you are going to answer the questions. Is first of all, how does the heterogametic sex differs from the homogametic sex? You have to tell me that. The second point that we are going to discuss online, and I'm going to ask questions randomly to the students. The second point is, what is the sexual phenotype of a fruit fly that has double X and triple Y, that is XX, Y, Y, Y sex chromosome and two sets of autosomes? The third one is, what is the phenotype of a person who has triple X and one Y chromosomes, which are actually XXX and Y sex chromosome. You have to like, I'm going to ask any one of you to like answer these questions. So all of you have to study it. And then in the last but not the least, you have to solve this problem. So let me just explain that to you. It's about hemophilia, which is reduced blood clotting. It is actually an X-linked recessive disease in case of humans. And the question is that a woman with hemophilia marries a man who exhibits a normal blood clot. So what you are going to like research about is, what is the probability that their child will have hemophilia. So you have to study them, and I'm going to discuss these questions with you in the class. So good luck.